Log on, tune in, find out. Another good idea from Cambridge.
Reducing bias in a class of methods for inferring species trees from gene trees.
Hi, today I'm going to talk about a method that's being carried out in collaboration with another student, Laura Helmkamp, at the University of Michigan, who's also Noah Rosenberg's lab. So as Louis Nachle talked about this morning, talked about this morning, the gene tree topology can differ from that of the species tree, even in the absence of events such as horizontal gene transfer and gene duplication. So here, for example, the blue tree has the same topology as the species tree, but the red tree doesn't. So when this occurs by chance, this is called incomplete lineage sorting, and it can complicate the inference of species trees from gene trees. So a number of methods have been developed to deal with incomplete lineage sorting. And there are many of them, but I'm just going to focus on these four today. So for example, the glass method was developed by Moselle and Roche and independently by Liu et al. Under the name of maximum tree. The shallow divergence method was presented by Madison and Knowles in the same paper in which they present the minimizing deep coalescence method, the stake method of Liu et al., and then the MAC method, which we'll talk about here and which will hopefully be presented sometime later this year. So all of these methods have the desirable properties that they're fast, and that they estimate both branch lengths and gene tree topologies. However, all these methods overestimate species divergence times. So today what I'm going to talk about is how we can reduce the bias in all of these methods. I'll focus primarily on the glass method because it's sort of representative of the other ones, and once you understand how to reduce the bias in that method, it's pretty straightforward to understand the other ones. So first, to see how the glass method works, consider the case of two taxa. So here, estimating the species tree just involves estimating the divergence time. So to do that using the glass method, we sample lineages from the taxa at a number of loci. At each locus, we compute the time to the first interspecific coalescent event at that locus. We do that for all loci, and then we take the minimum of these times to estimate the divergence time. Now, for multiple taxa, we do pretty much the same thing. We infer gene trees. We then consider each pair of taxa independently, and we find the time to the minimum interspecific coalescent event. We then do that for all pairs of taxa to generate a distance matrix, and then the species tree is estimated by applying single linkage clustering to this distance matrix. So note that when we assume that there's no exchange of genetic material between populations since the divergence time, then interspecific coalescent events necessarily happen more anciently than the divergence time. And so the glass estimate always overestimates the divergence time. In particular, it overestimates the divergence time by a quantity we'll call V, which depends on the number of lineages sampled in the present and on the divergence time. And when the divergence time is large, or when the number of lineages sampled is small, then this estimate can be a fairly large overestimate. So it's of interest to correct it. So a straightforward way to do that, would be to compute the expectation of this quantity V and simply subtract it from the glass estimate. The problem is that V depends on the divergence time, which we don't know, so we can't do that. So what we do instead is we treat the glass estimate as a random variable. We let T star be the observation of the glass estimate, and we find the value of the divergence time for which the observed glass estimate is equal to its expected value. And the solution to this equation is much less biased than the glass method, at least under the multi-species coalescent model, and so we call it the improved glass method or the iglass method. So in order to compute this expectation, we can write it in the following way as the divergence time plus the expectation of the quantity V. To compute this quantity, we first consider the case of a single locus. So let NA and NB be the numbers of lineages sampled in the present, and let KA and KB be the number of lineages remaining at the top of each branch at this particular locus. So by conditioning on Ka and Kb, we can compute this expectation, which was done by Takahata in 1989. In particular, computing this quantity here is a little bit tricky. Some closed form expressions for terms in this expectation were derived by Noah Rosenberg. And this probability that K lines remain at the top was derived by Tavare and others a long time ago. So then we sum over all values of Ka and Kb to get this expectation. Now, extending this to multiple loci basically just involves summing over all loci. This expectation is a pretty straightforward generalization. It involves some order statistics, and that's about it. The problem is that it's very time-consuming to compute. So we make the following approximation. Normally, under the coalescent model, we think of lineages as discrete quantities, discrete and random quantity. However, Maruvka et al. Showed that the number of lineages as a function of time is quite well approximated by its expectation. So we can make an approximation in which we assume that the number of lineages as a function of time from each population is just given by its expected value. However, we treat the interaction among lineages in the ancestral population as random. So here, using that approximation, we can derive the survival function of this overshoot v. And so here I'm showing the exact survival function and the approximation for, this is just for the case of one locus. On the x-axis, we have time in coalescent units, and on the y-axis, we have the distribution. And as you can see, the approximation is very close to the true value, and it only gets better as we add more loci. So I stole this diagram from Mariah. I thought it was a good use of arrows. So, OK. So to summarize, computing the eyeglass correction, we first compute the observation of the glass estimate. We then find the divergence time for which the observed value is equal to expected value, and the solution is what we're taking to be the eyeglass estimate. So to see how well this actually works in practice, we can first look at the bias in the glass method itself. So here I'm showing bias in the glass method for the case of estimating the divergence time between two taxa using 5 loci. So on the y-axis, we have divergence time increasing from 0 to 4. On the x-axis, we have increasing numbers of lineages sampled from the two taxa. Here, for the case of one lineage sampled per taxon, the bias in the glass method is constant at 0.2 for all divergence times, and that's because we're just taking the minimum over 5 exponentially distributed random variables with mean 1. OK, for low divergence times, increasing the number of lineages decreases the bias. But that's not the case for large divergence times. And the reason is that as the divergence time increases, so does the probability that one lineage remains at the top, in which case we're just back to this limiting case of one lineage sampled per taxon. So to see how glass and eyeglass compare, we can look at the ratio of the bias in the eyeglass method to the bias in the glass method. These are for exactly the same simulations. Here, for most of the parameter space, the eyeglass bias is considerably less than the glass bias, except very, very close to 0. We don't allow negative eyeglass estimates, and so the improvement isn't quite so dramatic right near 0. Similarly, mean squared error is about half that of the glass method, except again, right near 0. And of course, we can apply this to larger trees. This is a 5-taxon caterpillar tree. And again, this is looking at the ratio of mean squared error in eyeglass to mean squared error in glass. And again, it's about half. So in summary for this particular part of the talk, the glass estimates of divergence times are biased upwards, but we can correct both the bias and mean squared error using this eyeglass correction. And we can speed this up quite a lot using an approximation due to Maruvka et al. So the glass method performs usually quite well when gene trees are perfectly inferred, but usually that's not the case. The problem is that when one or more gene trees are inferred to have an interspecific coalescence time that's lower than the divergence time, then the glass method starts to run into problems. The other 3 methods here are much more robust to that problem, as we'll discuss. So all these methods work in approximately the same way. So in particular, the glass method starts by taking the minimum interspecific coalescence time across at a particular locus, and then takes the minimum over loci. The SD method begins by taking the minimum interspecific coalescence time at each locus, then taking the average over loci. Stake is a little bit different. It takes the average interspecific coalescence time at each locus, takes the average over loci. MAC takes the average interspecific coalescence time at each locus and takes the minimum over loci. So just to specifically discuss how each of these works, I'll discuss this as follows. So first, the shallowest divergence method. Again, like the glass method, we first find the time to the first interspecific coalescence at each locus. We do that for all loci, except unlike in the glass method, where we take the minimum of these times, we take the mean. Okay? So as you can see, this should be a little bit more robust to problems where gene trees are incorrectly inferred because we're averaging instead of just taking the minimum. The stake method is, again, very similar to these methods. So the stake method, however, at each locus,
instead of taking the minimum interspecific coalescence time, we find the average time of a coalescence involving a lineage from taxon A and a lineage from taxon B. And so then we do this for all loci. And then we take the average of these times as our estimate of the divergence time. So then finally, the MAC method, again, as in the stake method, we take the average interspecific coalescence time at each locus. Okay, however, instead of taking the average of these times, we take the minimum of these times. And this method has kind of the nice property that even when you don't make any corrections at all, this is still a consistent estimator of the divergence times as the number of loci increases. And so we can use that to also show that it's a consistent estimator of the species tree topology under the multi-species coalescent model again. So all of these methods can be corrected in a way that's similar to the correction for the glass method. So specifically, again, if T star is a particular estimate of the method, we find the value of the divergence time for which the particular estimate is equal to its expected value. So for most of these methods, the computation is very similar to that of the glass method. For the SD method in particular, this expectation is almost identical. For the MAC method, or for the stake method, where we're taking averages everywhere, this can be just shown to be the divergence time plus 1 coalescent unit. And then for the MAC method, this expectation is a little bit more complicated, and you can find it either by summing over topologies, which is quite slow, or you can find it by making an approximation that's very similar to the approximation that we made for the glass method. So again, we can look at the bias in all of these methods. So here again, I'm showing bias in all of the methods for the case of 2 taxa and I believe 2 sampled lineages per taxon. So in each heat map, as we move down the y-axis, we have increasing numbers of loci and increasing divergence time on the x-axis. We already looked at the glass method, but as you can see, in all of the other methods, the bias after making these various improvements greatly reduces the bias in all of these methods. And we can show also that the mean squared error is also greatly reduced. Again, this is under this particular model. So in summary, all these methods are pretty fast, and they estimate both topologies and branch lengths. However, the estimates produced of divergence times are all biased upwards, but we can correct that bias using these corrections that we derived. So I'm much earlier than I thought. I kind of sped through that. But I'd like to thank my collaborators on this project, Laura Helmkamp, who's another student in the lab who's been working to correct these methods. And we're both students in Noah Rosenberg's lab at the University of Michigan. So I'd like to thank some other people in the lab, Mike DeGiorgio and Lucy Huang, who in particular suggested the name eyeglass. The rest of the Rosenberg lab, the conference organizers, Newton Institute, our sources of funding, and thank you all very much for listening. So.
Questions?
Yes.
So I tried to run glass before, and the problem I ran into is that overestimating. It's actually that as you increase the number of loci, you start getting zero distances between pairs of sequences, and that will immediately make glass put the divergence times at 0.
Yep. So
how do we estimate these coalescent times, or how does this?
Okay. So one approach would be to take a method that's very different from the glass method. So in particular, you could estimate. You can estimate these times in the beginning by some maximum likelihood approach, so or Bayesian approach. So even if you were to have zero divergence, zero distance between sequences, you won't actually get zero divergence time. So that would be one way to get around that problem. But as you're suggesting, just throwing out those times sounds like a good idea. So I mean that may be one way to get around that, and then to correct those times subsequently, you could take that into account in these types of derivations that you're throwing away the minimum 20, so you derive the 20th order statistic, the 20th percentile order statistic rather than the first. So that would be one way to get around it. But yes.
So the other thing that happens when you look at lots of different genes is they can have different overall rates of evolution.
Yeah.
So in a
yeah.
Bayesian setting, you can handle that by estimating those things. And
Yep.
the shared species tree helps you do that.
Yep.
Do you see any possibility of doing something like that?
Of doing that here. I don't see a really easy way to do that here, given that you're constructing the species topology after the fact. No, I'm not really sure how you do that here. Yeah, sorry, but that's definitely an important issue with this type of method. Yep. Maybe we can talk about it later. I'll have to think about
Ideas,
that one.
but, maybe.
Okay, fantastic. Yeah.
No other questions? Thank you again.